right, kids, this is the second lecture for the DNA unit. We're going to be talking about transcription and translation in a little bit more detail today. But first off, let's just really quickly review about DNA and RNA. Some things that you should keep in mind here. There's a nice little Venn diagram that'll help you kind of separate things. First of all, some similarities. Remember, they both have, oops, I shouldn't be putting the similarities there. They both have nucleotides. That is the monomer for a nucleic acid. They both carry genetic information. So that's kind of important. It's very important. They also have some of the similar bases. Remember, a nucleotide has a phosphorus, a sugar, and a base. And so bases that they have in common would be adenine, cytosine, and guanine. Some bases they have different than DNA would have thymine, RNA has uracil. Think about what the D stands for in DNA. Remember, this is deoxyribonucleic acid. So in DNA, our sugar in the nucleotide is deoxyribose sugar. If you recall that from the other day. While in RNA, R stands for ribonucleic acid, and that's because in RNA, the sugar is called ribose sugar. We know that DNA is double-stranded, so it's a double helix, whereas RNA is just a single helix. Some other things, DNA is in the nucleus only. Remember, it cannot leave the nucleus. And so that's a problem since DNA has all the information to control our cell. If here's your cell and here's your nucleus and here's all the instructions inside, what happens if you need to do a job like out here at, say, a ribosome? Well, since the DNA can't get there to tell it what it needs to do, that's when we use this RNA, particularly mRNA. Because RNA can leave the nucleus, so it can leave the nucleus. And that's very important. There are three types of RNA. There's mRNA, which we just mentioned, that's messenger. So that carries the message from the DNA here in the nucleus out to wherever it needs to go in the cell. We also have ribosomal RNA, which we'll see in a little bit. That's just part of a ribosome. Remember, ribosomes are responsible for making proteins, and since DNA codes for making proteins, it's going to be very important. And the last kind is tRNA, which stands for transfer RNA. So you can pretty much imagine what kind of a function that's going to serve, but we'll get to more of that a little bit later. Some other things for DNA and RNA, similarities, going along with the nucleotides, they're each going to have a phosphate and a sugar, and we already talked about what the difference was between the sugars. DNA has deoxyribose, whereas RNA has just ribose sugars. So they have a phosphate, they have a sugar, and then remember the third part is the nitrogenous or nitrogen base. So those would be your A's, your T's, C's, G's, and U's, are the nitrogen bases. Okay? So transcription, remember we talked about transcription briefly the other day. There's an enzyme that's involved in this process called RNA polymerase. And remember, if we look at the word transcription, remember I keep emphasizing to you guys the importance of looking at the word and thinking about what it means. Well, if you look at this little part right in here, script, think about what a script is. Okay, like in a play, it's got all the lines in it, and even smaller than that in here, script or scribe means to write something down. Like a scribe is somebody who records notes on things. So transcription is this process of writing down the information. Specifically, again, if we draw our cell, if here's our nucleus that has all of our DNA in it, if we need to send that information to out in the cell, we first have to write it down before it can get sent there. So this transcription happens in the nucleus, and it's the process of writing down the information from the DNA onto mRNA so it can go out to the cell where it needs to do its job. There's a lot of terminology here that you don't really need to know, but I want you to just focus more on the diagram here. So here's this RNA polymerase, and it's reading along, well, it's going to here in a second. It's going to insert itself into the DNA molecule that it wants to copy down the information from. And so as you can see here, it's reading along this red strand. So the red strand is getting read as this guy moves in this direction. What happens then is the RNA polymerase takes free bases that are just kind of floating around the nucleus, and it base pairs them to the DNA. So for example, if this goes T and then A and then C and then G, on the RNA, we're going to base pair this. So T would give us an A on the RNA. A would give us a, remember, for making RNA, T bonds to, or sorry, A bonds to a U, C would bond to a G, and G would bond to a C. So if you notice, it's reading this red strand, and it's assembling a complementary set of bases here. Okay, it's not copying this TACG from the red strand, it's assembling a complementary set right here. Interestingly enough, if we were to look at what's on the blue strand up here, it'd be A, T, G, C, because this blue strand complements the red strand. So A and T, T and A, G and C, C and G. Again, notice there's a T here because this blue strand is DNA, whereas this light blue one is RNA. So once it gets done reading as much as it needs, this little segment of RNA is going to leave, and that's where we go out of the nucleus to wherever it needs to go in the cell in order to do its job. So that's transcription. So if we do a little bit of practice here, here we've got a template DNA strand. So you can tell it's DNA because it's got T's in it. And here we're going to start out by making a complementary DNA strand. So going from the template to a complementary strand, you know, if we were making a copy, that'd be called replication. We already talked about that the other day, so think about what enzymes are involved there. So if we do this one, go ahead and pause the video, and I want you to practice this. And here I'll give you the answers. So A goes to T, G to C, and then A, and then it'd be G, C, A, T, C, C, G, C, A, G, T. Okay, pretty easy. We've done this quite a bit. So now we're going to make RNA. So this process of making RNA, and again, we're going to read from our original template strand. So just so we're, you know, this is the format that we'll use in this class. So whenever you are making RNA, you'll make it from this template or original DNA strand. And so this process is called transcription then. So we're going to look to the top line here. So just pretend that this middle one's not here. A would go to a U, G would go to C, and T to A. We keep going. This would be G, C, A, U, C, C, G, C, A, G, U. So again, we base paired to this top line up here. Now, some of you may be pretty quick on this and picked up something. Since we base paired the complementary DNA and the RNA to this original strand, if you look, they're almost exactly the same. The only difference between them would be instead of having a T on this RNA strand, we have a U, because remember, RNA does not have T's, it has U's. Okay, so since we base paired both of these up to here, they should be the same. Let's do another example down here. So I want you to go ahead and look at this, pause the video, and go ahead and try to solve that on your own. All right, so if we do this really quickly, it's going to go A, T, G, and then C, T, G, 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 A, G, C, T, A. The RNA strand, we're just going to go through, and this is going to be A, U, G, and then we got C, U, G, and then G, G, A, G, C, and finally U, A. Okay, pretty easy. So if we look a little bit more at what exactly RNA does, mRNA in particular, It's made by RNA polymerase. That's an enzyme that we already talked about. So we already know what RNA polymerase does as it's reading one side of the DNA strand. So that's important. If you look right here, we're making this RNA strand right here, and it's being read from this strand of the DNA. We don't copy both strands, we only copy one. And there's a lot of regulation that goes on which side of the DNA we actually end up copying. It carries instructions from the DNA to the ribosomes for making proteins. Remember from our unit on cells that ribosomes make proteins. So this RNA strand, this mRNA strand, is going to come down here, and what's going to happen is it's going to feed through this ribosome. So this purple blob thing is a ribosome. So it's going to read through this ribosome. The ribosome is going to look at the bases that are on this mRNA, and it's going to assemble. AA stands for amino acids. You should remember that the monomers of a protein is an amino acid. So you've got kind of this assembly line effect going on here where we're hooking together these amino acids, which will eventually make a big long protein. So the ribosomes read the mRNA and assemble the new proteins out of amino acids. So we're going to take a little bit more look at this process here in just a second. All right, so if this is a strand of, well, let me ask you, is this a strand of mRNA or DNA? Hopefully you said RNA, because if you look, there's U's here. So that should tell you that this is RNA. So we've written down our message during transcription. So what do we do now? Well, we have to have some way to read this. Okay, just like if you were to, let me do this real quickly here. If we were to write a sentence out, so let's just write out a sentence. Biology is my favorite subject. And that's true. At least it should be. So if we read this sentence, notice I didn't put any spaces in here, right? But I mean, you can read this because you know how to separate these words out. Well, a ribosome does much the same way. I mean, here we've got this big long sentence of mRNA, and the ribosome, since it's going to read it to assemble the proteins, it has to have a specific way of doing this. Remember, we did that coding activity, and we talked about how important it was that we have a consistent code. Well, on RNA, we separate our letters here into groups of 3. And so there'd be a few more here, so we'll just add a couple more. So we separate it out into groups of 3 because these groups of 3 are like the words, just like biology is a word, is a word, my is a word, favorite is a word, and subject is a word. Here, each of these groups of 3 represents a word, if you will, to the ribosome, and those are called codons. Okay, so codon is a grouping of 3 nucleotides on the mRNA. They code for one amino acid. So ACG here, this is the first codon. It codes for a single amino acid. So these 3 letters together, the ribosome reads that, and it tells it what amino acid it needs to put onto the protein. Again, they're located on the mRNA. Very important. Codons are on the mRNA. There are 64 different combinations, and that should kind of make sense if you think about it. Since this is 3 letters long, so 1, 2, 3, and each of those 3 letters could be either A, C, G, or U. If we take that, that's 4 times 4 times 4. That would give us 64 different combinations of our alleles. Or sorry, not of our alleles, of our nucleotides. So there's 64 different words that could be made using these 4 letters grouped in groups of 3. They also tell us when to start and stop making proteins. So this strand of mRNA here has groups of 3 nucleotides called codons. They code for an amino acid, only one, and they also tell us when to start and stop making the protein. So these obviously code for amino acids, and we figured out this tool that helps us decipher what amino acid we're talking about. So for example, if we look here, let's say that the code on the mRNA was CAA. Well, this little wheel, and again, this is only for using on mRNA. You'll see why I stress that in just a minute. So this is for mRNA, or for looking at codons, we look at this wheel. CAA. I start at the center of my wheel. So I start and I work my way out. So I look at the first letter. That's C. My second letter is A, so I'm going to go here to A. And then my third letter is A, so I'm going to go to here. So CAA codes for the amino acid glutamine. So if a ribosome reads this codon, it's going to put glutamine onto the protein. This table, you might see one like this from time to time. When I learned how to do this in high school when I was your age, this is the one that I used. This one works very similar. The left side is your first base in the codon. The top is the second, and the right side is the third. So again, if we had C, A, A, I'd be looking at this row because that's my first amino acid, or sorry, my first nucleotide. I'd be looking at this column because that's my second one. So there's the A. And then I would go to this smaller row because that's an A. And GLN, if we come down here, glutamine. So it works the same way, it's just laid out a little bit differently. I think this one's probably a little more user-friendly, and this is the one that you'll use in my class. So if we do a little bit of practice here, oops, didn't mean to do that. We do a little bit of practice. GAU, that'd be aspartic acid. ACU, that'd be threonine. CUA would be leucine. And UUG would also be leucine. So pay attention to that for just a second. Each codon, so like UUC, only codes for phenylalanine. Just like UUA only codes for leucine. But if you look, UUA codes for leucine, UUG also codes for leucine, CUU, CUC, CUA, CUG, those all also code for leucine. So each codon can only code for one amino acid, but some amino acids have several codons. Okay? All right, so what you just did unknowingly was the process of translation. So think about what the word translation means. If I were to write a sentence to you in Spanish, you might have to translate it into English. So turn it into something usable or something that you can understand. Well, the ribosome is responsible for translating. So our coded message here on the mRNA enters the ribosome. The ribosome reads the mRNA one codon at a time. So it's like translating a sentence one word at a time. And then we have this other kind of RNA called tRNA. Remember, T stands for transfer. It brings in the appropriate amino acid. So if you think of the ribosome as like the chef, and the mRNA is like the recipe, the chef reads the recipe or the list of ingredients one at a time, and he yells out, "Oh, I need flour." And his little helper, the tRNA, goes and gets flour and brings it to him so that he can add it to his pot of whatever he's making. So here, the ribosome, this blue thing, is reading a codon, AUG, one codon at a time. So there's one codon, here's the next codon, here's the next codon. And it's saying, okay, AUG, I need methionine. And so the tRNA brings in the proper amino acid. If you look here, GCC, well, let's go back to our little wheel here. GCC should be alanine. And look, the next tRNA is bringing in an alanine. So it'll come in, it'll hook up right here, and this alanine will get attached to the methionine. You can see that happening right here. Once that happens, this guy goes to find another methionine, and we read the next codon. So UGG, that'd be tryptophan, and then ACU, that'd be threonine. So reading this one codon at a time, the ribosome feeds the mRNA through, so it's going to move that way, and it reads it and assembles the amino acids. And eventually you'll get tons of these, thousands maybe, of these amino acids hooked together to make a protein. So the ribosome attaches the amino acids to form that polypeptide or the protein. If we look a little more about tRNA, this is obviously a little bit caricatured. tRNA looks a little different. I don't know if I have a diagram on here or not, but its overall shape is like a T, which is convenient because T stands for transfer, and it also looks like a T. So again, here's our mRNA strand. Think about what this group of 3 is called on the mRNA. Okay, it's called a codon. So here's our tRNA. This particular one has leucine attached to the other end. That's an amino acid. This one has alanine at its other end. That's an amino acid. I want you to pay special attention to right here. There's a group of 3 nucleotides on the opposite end of the amino acid on this tRNA. And if you look, C, G, U, A, U. Those 2 groups of 3 are complementary. They complement each other. These are base pairing up. I mean, it's not like the ribosome has a brain and it can read this message and say what it needs, but the reason why the correct amino acid gets put in place is this tRNA has a group of 3 that will complement a group of 3 on the mRNA. If we look here, G to C, C to G, and G to C. This is how we know what amino acid to bring in, is these tRNA have a complementary group of 3 that matches up with a codon on the mRNA. So the transfer amino acid brings in the tRNA transfers the amino acid from the cytoplasm to the ribosome. So you just got amino acids floating around in your cells. You get a lot of those from your diet, some of them your body makes. And the tRNA brings them to the ribosome to be attached to the protein. The tRNA, this group of 3, it's called an anticodon. It's called an anticodon because it's complementary to, or it's the anti-nucleotide compared to the codons on the mRNA. And they have one amino acid at the other end. So for example, this guy, as soon as he gets rid of his leucine, he's going to leave the ribosome and go find another leucine. That's the only one that he'll bind to. This one, as soon as he adds his alanine, he'll go find another one. So you've got dedicated tRNAs that go and find one specific amino acid, and then that's why their code works, because this codon will attach, anticodon will attach to a codon on the mRNA, and it brings in only one amino acid. This is what the tRNA actually looks like. If you look here, you can kind of see this T. This is a big, long strand of molecules that makes up the tRNA. tRNA is fairly complex. But we also have codons on our mRNA that tell us when to stop or start and when to stop. So for example, the start codon, and you'll want to write this down because you'll need to know this, is AUG. It codes for methionine. So methionine is the first amino acid that we start with anytime we make a protein. So if we look on this code here, AUG, it's going to give us methionine. So that tells us to start making an amino acid, making a protein from the amino acids. We also have codons that tell us when to stop. Okay, obviously at a certain point you'll need to stop making a protein. And those codons are UAA, UAG, and UGA. Excuse me. So if you look over here in our diagram, UAA is a stop, UAG is a stop, and UGA is a stop. So as soon as the ribosome reaches one of those 3 codons on the mRNA, it's going to stop making the protein because our protein's done. So let's put this all together real quickly. I want you to pause the video and copy down this table. I gave you the DNA strand, so you can think of this as the template. And I want you to go ahead and fill in the mRNA, the tRNA, and the amino acids. Notice I'm not having you make a DNA strand here. So make the mRNA, the tRNA, and the amino acids. Remember, the mRNA is base paired to the DNA. The tRNA base pairs to the mRNA. And we use our wheel to get the amino acids from the mRNA. So go ahead, pause the video, and I want you to do that while I fill it in. Okay, welcome back, and hopefully you were able to fill this out. Again, so matching this up, the DNA is what we read to get the mRNA. So T goes to A goes to U, C goes to G, and so on. The tRNA, these anticodons are complementary to the codons. So A goes to U, U goes to A, G goes to C, and so on. And to get the amino acids, remember we read from the codons on the mRNA. So we're going to be using this wheel with the codons from this row, okay, the mRNA. This is probably the biggest mistake that students make when it comes to the test, is that they read the tRNA instead of the mRNA. We use that wheel with the mRNA only. So AUG is methionine, GCG goes to alanine, GAU is aspartic acid, UCC is serine, CGU and AGG are both arginine, and UGA is stop. So go ahead and if you need to pause the video, you can, and check your work to make sure you did it correctly. And you can practice this one on your own. So if you need to pause the video to get it, go ahead. You can do this one on your own. Last thing I want to talk about is mutations. A mutation is just a change in the genetic material. So that means if we're looking at DNA or RNA, it's called a mutation if we change the nucleotides that are there. So if instead of being AGG, it becomes AGC, that would be a mutation because it's changing the genetic material. A point of information, anything that changes the genetic material is called a mutagen. So for example, a common one is ultraviolet light. When you go out in the sun, you know, there's UV light. You always hear about how you need to wear sunscreen to protect from UVA and UVB rays. Well, ultraviolet light can actually cause changes or mutations to your DNA. Now, that doesn't mean you're going to grow a third arm or you're going to, you know, be a cyclops or any crazy mutation that you might think of. But it can have some negative impacts. Remember that since DNA and RNA they code for making your proteins, if you change that, you know, you might put in the wrong amino acid and it might change the shape of your protein. And that can be very, very bad. There are certainly several diseases that we'll look at that result from changes or mutations to the DNA. So there's two kinds of mutations, and one is called a point mutation. Point meaning one spot. It's a change in a single nucleotide. So for example, if my DNA was supposed to say CGC, but then a mutagen comes along and changes it to CAC, that would be a point mutation. Okay, we only changed the middle letter here in the codon. Okay, the alternative would be what we call a frameshift mutation. Now this can be either what we call adding or insertion or deleting a base pair. So for example, if we have CGC ATC and then our mutagen comes in and we get CGA CATC, we had something happen here. Can you figure out what it is? Same thing down here. CGC-ATC becomes CCA-TC. Well, obviously on this one, we cut out that G. So that would be a deletion. The one above would be an insertion. Because if you look carefully, this goes CGC. Here we go, CGA-CAT. So we inserted an A between that G and C. So this is the mutation right here that was inserted. Now, what's really important on a frameshift mutation is because your ribosome reads the RNA 3 at a time, if I insert or delete a base, it shifts everything after it. Okay, think about like if you were in line, you know, in line at class, and I said every third person who comes into the room gets $100. And you're supposed to be you're person number 9 in the row, so you would get, you know, $100 if you were, you know, the 9th person into the room because you'd be a multiple of 3. Well, what happens if your friend Bob cuts in line in front of you? Well, now it shifts that group of 3. So everybody after Bob, now they've changed their number. So you know you miss out on $100. Now I'm not going to give you $100, but in our DNA, if you do that, now instead of reading ATC, our ribosome is going to read CAT, and that could change the amino acid and actually change every amino acid after it. So frameshift mutations are particularly nasty. So here's an example of frameshift mutations. If this is our original mRNA, so AUU, CGU, AGU, GCG. Let's look at the amino acids that we get here. So AUU. AUU would be isoleucine, so we'll just write iso right here. CGU. CGU would be arginine. Oops, wrong way. So this would be arginine, I'm just abbreviating. AGU. AGU would be serine. And then GCG, GCG would be alanine. And then, it would go on from there. So there'd be, more codons that would code for more amino acids. Well, now let's look if we have a mutation here. And it's red A. That's where the mutation happened. If you notice, it shifted everything over. So here the U is shifted over, the A is shifted over, so on and so forth. So if we do this, AUU is still going to be isoleucine. CGA is going to be CGA is arginine. So that one's still okay. If you notice that, changing the third base in the codon actually didn't change this amino acid. Think about if you change the first one or even the second one. Usually a change to the third isn't a big deal. But I mean, it can be, but sometimes you'll get the same amino acid. UAG, hopefully some of you remember that one, but UAG is a stop. So that means our amino acid stops right here. That could be really bad. If maybe the one right before this was methionine, now you've got a protein, quote unquote, that's only 3 amino acids long. That's probably not going to do what it needs to. So this shift can cause us to change the amino acids, and that could prematurely stop our protein, which could be very, very bad. So that's pretty much it for transcription and translation. You'll have an assignment that you can do some practice on.